Good morning. The chapter for today is Cell, the structural and functional unit of life. Let's see the subheadings of this chapter. What is a cell? Discovery of the cell, cell theory, prokaryotic and eukaryotic cell shape, size and number, structure of a typical cell. The other subheadings are in the next slide. Cell organelles found in cytoplasm, nucleus, protoplasm, differences between a plant cell and an animal cell, cell functions, an overview of cell. What is a cell? A cell is a structural and functional unit of all living beings. Any function performed by organism is the outcome of the activities of its cells. Many cells come together to form a tissue, and tissues collectively form organs. Thus, every organ in our body, like skin, muscles, brain, and heart, is made up of hundreds of thousands of cells. A cell is microscopic, that is very small. It is the smallest part of the body of an organism capable of independent existence. All new cells arise as a result of division of pre-existing cells. All living beings start their life as a single cell. Every cell has its own lifespan. The old and worn out cells are continuously replaced by new cells. Each cell consists of an organized mass of protoplasm surrounded by selectively permeable covering called plasma membrane. In an animal cell, the plasma membrane is the only limiting membrane. However, in plant cells, fungi and bacteria, a cell is further surrounded by a cell wall. Discovery of the cell. The invention of the microscope helped in the discovery of the cell. The first microscope was constructed by Anton von Leeuwenhoek. It consisted of a single biconvex lens and was known as simple microscope. In this microscope, the eyepiece was located close to the lens on one side, while the object was mounted on the screw point on the opposite side of the lens. Some of the Leeuwenhoek's microscope could magnify objects up to 200 times. Robert Hooke developed a compound microscope by using two lenses for achieving greater magnification. In such a microscope, the object to be seen was placed on a stage below, and the light coming from an oil flame passing through a water flask was shown on it by a convex mirror. Cells were discovered by Robert Hooke in 1665. He studied a thin slice of cork under his microscope. He observed that the cork slice had a large number of compartments joined together in a honeycomb-like structure. He named these compartments as cells. The light microscope. The ordinary light or optical or compound microscope used extensively in laboratories these days is a fairly improved design of Hooke's microscope. It consists of two lenses, the eyepiece lens and the objective lens, which are combined to produce a greater magnification. The light microscope has a magnification up to 1500 times, good enough to see cells, larger organelles, and bacteria. The electron microscope. Invention of an electron microscope unfolded many new discoveries about cells. The electron microscope has a power of magnification over 2 lakh times and resolution much greater than those of an optical microscope. An electron microscope can resolve points 1 nanometer apart. In this microscope, a beam of electrons is passed through the section of specimen material to produce the image. The electron beam passing through the specimen section is focused by electromagnets and is projected on a fluorescent screen for direct view onto a photographic plate for permanent recording. The resulting photograph is called an electron micrograph. Cell theory. The cell theory was formulated by two German biologists, Matthias Kalden and Theodor Schwann. Their theory stated cell as a basic structural and functional unit of all living beings. In 1855, Rudolf Virchow added a phrase, omnis cellula e cellula, meaning all cells arise from pre-existing cells. In context of modern researches, the cell theory can be stated as follows. Cell is structural and functional unit of all living beings. All living organisms are composed of cells. All new cells arise as a result of division of the pre-existing cells. A cell is the smallest unit of all the living organisms. Prokaryotic and eukaryotic cells. On the basis of their nuclear organization, cells have been classified into two types, prokaryotic cells, eukaryotic cells. Let's see the prokaryotic cells. Pro, which is before, karyon, nucleus. Prokaryotic cells means cells having primitive nucleus. The nuclear material in this type of cells is not enclosed by a nuclear membrane. The genetic material is equivalent to a single molecule of DNA. It does not form chromatin. These are single-celled and do not have well-organized nucleus. These cells lack membrane-bound cell organelles like mitochondria, lysosome, endoplasmic reticulum, chloroplast, and nucleolus. Bacteria and blue-green algae are examples of prokaryotic cells. Eukaryotic cells. EU, true, karyon means nucleus. Eukaryotic cells means cells having a properly defined nuclear membrane. In these cells, the genetic material is made up of two or more DNA molecules, which form chromatin fibers. The nuclear material is enclosed in a nuclear membrane. These cells have a well-organized nucleus and have developed membrane-bound cell organelles such as mitochondria, endoplasmic reticulum, lysosome, chloroplast, and nucleolus. Eukaryotic cells occur in plants, animals, fungi, and protozoa. In this slide, let's see the differences between prokaryotic and eukaryotic cell based on the features like size of the cell. In prokaryotic cell, it is small in size, that is 1 to 10 micrometer. In eukaryotic cell, it is large in size, that is 3 to 100 micrometer. Nucleus have undefined nuclear region due to absence of nuclear membrane. In eukaryotic cell, have well-defined nuclear region surrounded by nuclear membrane. Chromosome. In prokaryotic cell, it is single molecule of circular DNA. In eukaryotic cell, it is more than one chromosome. Nucleolus. It is absent in prokaryotic cell. Nucleolus is present in eukaryotic cell. Membrane-bound cell organelles. Membrane-bound organelles are absent in prokaryotic cell. Membrane-bound organelles are present in eukaryotic cell. Cell shape, size and number. Cell shape. Cells show a great variation in their shapes to suit their functions. Most cells have a definite shape. Cells may be spindle-shaped like muscle cells, elongated nerve cells, oval red blood corpuscles, cuboidal germ cells, branched osteocytes and chromatophores, and so on. Some cells may not have definite shape, that is, they have irregular shapes like amoeba and leukocytes or white blood corpuscles. Cell size. Smallest. The smallest known cell is mycoplasma or PPLO, pleuropneumonia-like organisms. Its size varies from 0.1 to 0.5 micrometer. The bacterial cell is 0.5 to 5 micrometer in size. Human red blood corpuscles are 7 to 20 micrometers. Human liver and kidney cells are 20 to 30 micrometers in size. Longest. Nerve cells are about 90 to 100 centimeters in size. Acetabularia, a single-celled algae, has a length of 10 centimeters. In plants, fibers are quite long. For example, 4 centimeters in cotton and 1 meter in hemp. Largest. The largest cells are bird eggs. The egg of an ostrich is 170 into 135 mm, which is largest single cell, and that of hen is 60 into 45 mm. Human female ovum is about 0.1 mm in diameter. Cell number. Single-celled. Many organisms are made up of only one cell. They are called single-celled or unicellular. For example, bacteria, amoeba, paramecium, euglena, chlamydomonas, etc. Multicelled. Most organisms are multicellular. That is, they are made up of several cells. Newly born human baby has 2 trillion cells, while an adult has about 100 trillion cells. Structure of a typical cell. Although cells of different organisms differ in structure, cells within the body of multicellular organism differ in shape, size, and functions. In spite of these differences, every cell shows some basic structure: cell membrane, cytoplasm, and nucleus. In the following diagram, we can see the ultrastructures of a plant cell and an animal cell. Cell wall. Cell wall surrounds the cell membrane. Cell wall is an outer rigid, protective and supportive covering of bacterial, fungal and plant cells. It lies outside the plasma membrane. It is made up of cellulose in case of plant cell. Cellulose provides mechanical strength and rigidity to the cell. Cell wall is not present in animal cells. Functions. It provides a definite shape to the cell. It protects cell membrane and internal structures from attack of pathogens and mechanical injury. It is freely permeable, allowing the substances to move in and out of the cells without any hindrance. It provides rigidity and mechanical strength to the cell. Cell membrane or plasma membrane. Every cell is bound by a thin, delicate membrane called the cell membrane or plasma membrane. It is the outermost covering of an animal cell, while second outer covering of a plant cell, which separates the contents of a cell from its outer environment. Plasma membrane, being very flexible, helps in taking in food and other substance from its external environment. This is known as endocytosis. Functions. Plasma membrane is selectively permeable, therefore it allows only selective substances to pass in and out of the cell. It bounds the semi-fluid contents of the cell. It protects the cell from injury and provides an outer boundary to the cell. It allows the flow of materials and information between different organelles within the cell as well as between one cell and another. Cytoplasm, the house of cell organelles. The space between plasma membrane and nucleus is filled by translucent homogeneous semi-liquid called cytoplasm. Several chemical reactions takes place in cytoplasm. It is living and always in a state of some movement. In the cytoplasm, various cell organelles are found floating. Functions. It helps in intracellular distribution of molecules, enzymes, and nutrients. It helps in exchange of materials between different cell organelles. Breakdown of glucose takes place in cytoplasm. Biosynthesis of nucleotides, proteins, and fatty acids takes place in cytoplasm. Cell organelles found in cytoplasm. Different types of cell organelles. Cell organelles are divided into membranous and non-membranous. Let's see the membranous. Membranous is further divided into double membranous and single membranous. Double membranous is further divided into endoplasmic reticulum, Golgi apparatus, mitochondria, and plastids, plant cell only. Single membranous is divided into lysosomes and vacuoles. Now let's see the non-membranous. In non-membranous, we can find ribosomes and centrosomes, animal cell only. Endoplasmic reticulum, ER, the skeletal framework of the cell. Endoplasmic reticulum is an interconnected network of double membrane lined channels distributed over the entire cytoplasm. The ER is connected with the cell membrane at its outer end and with the nuclear membrane at its inner end. Endoplasmic reticulum is of two types, smooth and rough. Smooth endoplasmic reticulum SER does not bear ribosomes. This type of endoplasmic reticulum is found in liver cells and muscle cells. Rough endoplasmic reticulum RER has a rough appearance because a number of ribosomes are attached to its outer surface. It is very well developed in plasma cells, fibroblasts, and goblet cells. Functions. Endoplasmic reticulum divides the cytoplasm into small compartments. It gives rigidity to the cell. Therefore, it acts as a skeletal or supporting framework of the cell. It helps in intracellular transport, distribution of material from one part of the cell to other parts. Hence, it is also known as circulatory system of the cell. It provides a large surface area inside the cells for various biochemical activities. Ribosomes, the sites of protein synthesis. Ribosomes are small granules found freely in the cytoplasm as well as attached to the outer surface of the rough endoplasmic reticulum. Functions. Ribosomes help in protein synthesis. Hence, They are called protein factories of the cell. Mitochondria, the powerhouse of the cell. Mitos means thread, chondrion means granule. Mitochondria are the double-walled cell organelles where cellular respiration occurs to release energy. Typically, mitochondria are sausage-shaped, but these may be granular, rod-shaped, spherical, or thread-like also. Each mitochondrion is covered by double membranous bag. Its outer membrane is smooth, while the inner membrane is outgrows into finger-like folds called cristae. The cavity of mitochondria is filled with matrix. The mitochondrial matrix contains lipids, proteins, circular DNA, and RNA. Functions. Mitochondria are miniature biochemical factories where foodstuffs are oxidized and energy is released. This energy is stored in the form of ATP, adenosine triphosphate. Hence, mitochondria are called the powerhouse of the cell. Golgi apparatus, the delivery system of the cell. The Golgi apparatus comprises very small vesicles of different shapes, generally located near the nucleus. Golgi complex contains three distinct components. They are flattened sacs or cisternae running somewhat parallel to each other, clusters of tubules and vesicles, and large vesicles or vacuoles. These membrane-bound vesicles many a times connect with endoplasmic reticulum. In most plant cells and in the cells of invertebrates, several units of Golgi complex are found diffused in cytoplasm and are known as dicytosomes. Functions. Golgi apparatus helps in secretion of mucus, enzymes, and hormones. The material synthesized near endoplasmic reticulum is transported to the various target sites inside and outside the cell through Golgi apparatus. It helps in storage of secretory products. Plastids, the kitchen of the plant cell. Plastids are cell organelles found only in plant cells. Depending on color, these are of three types: chromoplast, chloroplast, and leucoplast. Chromoplast, chroma, color, plasto, formed. They contain fat-soluble yellow, orange, or red-colored pigments. These are mostly present in petals and fruits. These provide color to flowers and fruits. These contain the following coloring pigments: xanthophyll, yellow-colored pigments; keratin, orange-red pigments. Chloroplast. Chloros means green, plasto means formed. They are green plastids. They possess photosynthetic pigments, chlorophyll and carotenoids. They form bulk of green parts of plant like leaves, young stems, etc. They trap solar light energy and absorb carbon dioxide for manufacturing food through photosynthesis. Chloroplast also contains other pigments, yellow and orange, but these are masked by large quantity of chlorophyll. Chloroplast contains DNA, RNA, and various enzymes essential for photosynthesis. Leucoplast. Leucos, white. Plasto means formed. These are colorless plastids. They have no pigments and are named on the basis of substance they store. They store starch. Cells of potato have a ton of large amount of leucoplast. Functions. The green plastids called chloroplasts help in photosynthesis and thus help in synthesis of food. These are called the kitchen of the cell. Chromoplasts are colored plastids that provide color to the flowers and fruits. Leucoplasts help in storage of food. Centrosome and centrioles in animal cell only. Centrosome and centrioles are found in animal cell only. A centrosome is small naked protoplasmic structure present near the nucleus. It consists of two small granules called centrioles, which lie at right angles to each other. During cell division, centrioles migrate to the opposite poles of the cell. Functions: they initiate and regulate cell division. They help in spindle formation during cell division. Lysosomes: lyso, digestion, soma, body. Lysosomes are membranous sacs budded off from Golgi bodies. The enzymes released by rupture of lysosomes help in intracellular digestion by destroying and digesting foreign substances around them. Hence, they are called suicide bags of the cell. Lysosomes help in intracellular digestion. They digest stored food substance during starvation of the cell. They bring about cellular breakdown and are associated with aging. They dissolve damaged cells. Vacuoles, non-living. Vacuoles are fluid-filled membrane-bound spaces in cytoplasm. In animal cells, vacuoles are small. In mature plant cells, the small vacuoles fuse to form single large central vacuole. Vacuoles are bound by a membrane known as tonoplast. They are filled with fluid called cell sap, which consists of water and variety of compounds. Functions: vacuoles help the cell to remain turgid. They play an important role in growth by helping in elongation of cells. They provide a watery environment for the accumulation and storage of water-soluble compounds, sugars, minerals, pigments, etc. In protozoans like amoeba and paramecium, vacuoles help in getting rid of body waste. Nucleus. Nucleus is a small round spherical mass, sometimes cylindrical, present almost at the center of a cell. It is the most important part of the cell. The nucleus contains nuclear envelope or nuclear membrane, nucleoplasm or nuclear sap, chromatin network or chromosomes, and nucleolus. Nuclear envelope or nuclear membrane separates the nucleus from cytoplasm. It is a double membrane structure and contains a large number of pores. The nuclear pores control the passage of substance from inside the nucleus to its outside, that is, cytoplasm or vice versa. Nuclear envelope is filled with transparent semi-fluid, dense nucleoplasm or nuclear sap. The nucleoplasm contains chromatin network and nucleolus. Chromatin network, that is chroma, which is color, is tangled fibrous mass of thread-like structures. During cell division, the chromatin threads condense and become ribbon-like to form chromosomes. Each chromosome consists of two similar threads attached to a centromere. They are composed of DNA, deoxyribonucleic acid molecules, and proteins. The number of chromosomes is always constant for a particular species. In human beings, the number of chromosomes is 46, 23 pairs. The garden pea contains 14 chromosomes, onions 16 chromosomes, maize 20 chromosomes, honeybee 32 chromosomes, while dog has 78 chromosomes. The information for inheritance of characters from the parents to the next generation passes in the form of DNA. Functional segments of DNA are called genes. Genes are the carriers of heredity. Each nucleus has at least one nucleolus. Nucleolus is a dense round structure attached to a chromatin fiber. The nucleolus may be one or more in number and is not bound by any membrane. It is rich in proteins and RNA, ribonucleic acid, molecules. It helps in protein synthesis by forming and storing RNA. Functions. The nucleus controls cell metabolism and other activities of the cell. Hence, it is also called the master or director of the cell. It regulates and coordinates various life processes of the cell. Chromatin part of the nucleus possesses all genetic information that is required for the growth and development of the organism, its reproduction and metabolism and behavior. It contains genes that are carriers of heredity. All variations are caused by changes in genetic material present in the nucleus. Protoplasm. Protoplasm is the living substance comprising cytoplasm and the nucleus in a living cell. The chemical composition of protoplasm varies from one cell to another. The common elements found in protoplasm are carbon, hydrogen, oxygen, nitrogen, iron, phosphorus, sulfur, etc., which constitute carbohydrates, proteins, fats, minerals, and water. All the living components of cell lie in the protoplasm and perform their functions. Differences between a plant cell and an animal cell. The following table shows us the difference between an animal cell and a plant cell based on the following features. Size. In animal cell, it is usually small in size. Plant cell, comparatively large in size. Cell wall. Cell wall is absent in animal cell. In plant cell, a definite cell wall made up of cellulose is present. Vacuoles. Vacuoles are either absent or, if present, are small in size and scattered in animal cell. In plant cell, a mature, usually a single large, well-developed central vacuole is present. Golgi bodies. In animal cell, Golgi bodies are well developed and present near the nucleus. In plant cell, the components of Golgi bodies are diffused in the plant cells and are called dicytosomes. Centrosome and centrioles. In animal cell, centrosome and centrioles are present. In plant cell, centrosome and centrioles are absent. Plastids. In animal cells, plastids are absent. In plant cell, plastids are present. Storage of reserve food. In animal cell, reserve food is stored in the form of glycogen. In plant cell, reserve food is stored in the form of starch. Cytoplasm. In animal cell, cytoplasm is dense and more granules. It fills entire cell. In plant cell, cytoplasm is not so dense. Only a thin line of cytoplasm is arranged on the periphery. Cell functions. Functions of cells in all living beings. Growth. Growth is a result of new cells being produced by cell division. Any substance added to increase the bulk of body of an organism is also produced due to the activity of the cells. Reproduction. No matter how an organism reproduces, whether sexually or asexually, it is again the cells that carry out the process. The male sperm is a cell, and so is the female egg. When you grow a new plant from a cutting, such as a rose or sugarcane, It is again the cells in the cutting that redivide and result in the growth of a new plant. Functions of cells in plants: absorption of water and minerals from soil. The root cells absorb water and minerals from the soil. The cells of stem conduct this water and minerals to all parts of the plant. Production of food, that is starch. The food starch produced by plant is through the activity of green cells of the leaf. The green cells contain chlorophyll, which traps sunlight for food synthesis. Produce colorful flowers, and the color of flowers and fruits is due to the pigments developed inside the cells. Opening and closing of stomata. In leaves or young stem, and opening of petals of a flower are all due to changes in the water content of their cells. Functions of cells in human beings. Movement. The movement of limbs or the body as a whole, that is locomotion, is the result of the contraction of muscle cells. Tasting food while eating. Taste of any food that you have is the result of taste cells, sensory of your tongue. Digestion of food and its absorption. The enzymes produced by cells of digestive glands digest food and intestinal cells absorb it. Transport of oxygen in the body. The oxygen supplied to the body parts is absorbed by the red blood cells from inside the lungs and then transported to all parts of the body. Thermoregulation. The body temperature is maintained by the activity of skin gland cells by sweating in summer or increased cell metabolism in winters. Let's see an overview of cell. Parts and organelles found in plant cells only. Cell wall. Key characteristics. An outer rigid protective supportive covering of plant cells only. It is mainly composed of cellulose. Key functions. Provides shape and rigidity to the cell. Freely preamble. Allows entry and exit of substances in solution freely. Protects plasma membrane and internal structures of the cell. Plastids. Key characteristics. Cell organelles of three types: chloroplast, chromoplast, and leucoplast. Double-layered, proteinaceous, contains DNA. Have matrix and disc-shaped grana which contains chloroplast. Key functions. Chloroplast, the green plastids help in photosynthesis. Chromoplast impart color to flowers and fruits. Leucoplast helps in storage of food. Organelle found in animal cells only. Centrosome. Key characteristics. A small naked protoplasmic structure present near the nucleus. Consists of two small granules called centrioles. Key functions. Initiates and regulates cell division. Helps in spindle formation during cell division. Parts and organelles common in both animal and plant cells. Cell membrane. Key characteristics. Outermost covering in animal cells. Next to cell wall in plant cells. Selectively permeable. Bounds the semi-fluid contents of the cell. Made up of lipid and protein bilayer. Has fine pores. Key functions. Allows only selective substances to pass through it. Protects the cell from injury. Maintains the shape of the cell. Cytoplasm. Key characteristics. The space between plasma membrane and nucleus filled with amorphous translucent colloidal liquid. Contains inorganic molecules, water, organic compounds, and various cell organelles. Key functions. Helps in exchange of materials between different cell organelles. All metabolic activities such as biosynthesis of proteins, nucleotides, fatty acids takes place here. Endoplasmic reticulum. Key characteristics. Interconnected system of membrane-lined channels that run through cytoplasm. It may be smooth or rough depending on the absence or presence of ribosomes on it. Key functions. Helps in the synthesis and transport of proteins and fats. Acts as a skeletal framework of the cell. Acts as a circulatory system of the cell. Ribosomes. Key characteristics. Small granules found either floating in cytoplasm or attached to the outer surface of rough endoplasmic reticulum. They are also found in mitochondria and plastids. Key function. Help in protein synthesis. Mitochondria. Key characteristics. Typically sausage-shaped bodies. Double-walled, outer membrane smooth, inner membrane outgrows into folds. Mitochondrial matrix contains lipids, proteins, circular DNA and RNA. Key functions. Oxidize food to release energy in the form of ATP. Synthesize respiratory enzyme and many amino acids. Golgi bodies in animal cells and dicytozymes in plant cells. Key characteristics. Stacks of flattened sacs or cisternae. Contain vesicles, vacuoles and tubules. Key functions. Help in secretion of mucus, enzymes and hormones. Help in storage, modification and packaging of secretory products. Help in formation of lysosomes. Lysosomes. Key characteristics. These are the membranous sacs budded off from Golgi bodies. Have a resistant membrane covering which protects the cell from digestive enzymes contained inside them. Key functions. Help in intracellular digestion. Provide energy during starvation. Bring about cellular breakdown during aging. Nucleus. Key characteristics. A dense spherical or cylindrical body present at the center of a cell. Enclosed by nuclear envelope which has pores to allow entry and exit of substances. Contains chromatin which contains DNA. Key functions. Regulates all functions within the cell. Contains chromatin fibers that possess all genetic information in genes. Help in cell division. Nucleolus. Key characteristics. A dense round structure attached to a chromatin fiber at nuclear envelope. May be one or more in number. Key function. Helps in protein synthesis by forming and storing RNA. Chromatin fibers. Key characteristics. A tangled fibrous mass which condenses to form chromosomes during cell division. Key function. Contain chromosomes which have genes to carry hereditary information. Vacuoles. Key characteristics. Fluid-filled membrane-bound spaces. In plant cells, vacuoles are large. In animal cells, vacuoles are small. Key functions. Help cells to remain turgid. Help in cell elongation. Help in the storage of water and other substances. Granules. Key characteristics. These are small particles, droplets or crystals. Key function. Serve as food for the cell. For example, starch in plant cells and glycogen in animal cells.